Bombay blood group is a rare blood group, and it was discovered in Asian population, particularly in the population, Bombay population. It was discovered by the renowned pathologists in Bombay. So what is this Bombay blood group? Normally, the blood groups are formed because of the A and B genes. Chromosome number 9 has got A and B genes, and there is an independent gene called as H gene. This H gene is dominant in every individual, so it will product the gene product. It will form the H gene product. Now, if some person has got A gene dominant, that will form A gene product, which will act through the H product to form A antigen on the RBCs, and the person's blood group will be A. Similarly, if a person has got B gene dominant, then B gene product will be formed. It will act through H product, and the person's blood group will be B. If both A and B are dominant, then both gene products will be formed and the blood group will be AB. If both A and B are recessive, then no A and B gene product and the person's blood group will be O. No antigens found on the RBCs. So that O is because A and B both genes are recessive, but H is still dominant. It has to be dominant in every individual. Having said this, now talk about the Bombay blood group. Let's talk about the Bombay blood group. In this population, very insignificant number, very less number of people have Bombay blood group. H gene itself is recessive. So that is very, very rare. And recessive gene means we will write it as small h. If the H gene itself is recessive, then no gene product can be formed. H product will not be formed. And if there is no H gene product, then it doesn't matter if the person's A or B gene is dominant, would not matter. Whatever gene is dominant, A or B, there is no H product. So there is no formation of A or B antigens possible. Remember the prerequisite, there has to be the H product. So if there is no H product, then A and B antigens cannot be formed, and therefore the person's blood group will be, it will be O. But only thing is, this time it is O because of H recessive. So it is commonly written as O small h. I mean, it's O blood group, but because of H gene recessive. What is the clinical significance of this? Clinical significance is that this person will harbor 3 antibodies in the plasma. Anti-A and anti-B antibodies, because O blood group has these 2 antibodies in the plasma, plus anti-H antibodies also will be present in the plasma. And therefore, this person can receive blood only from another Bombay blood group individual. The safe blood transfusion can occur only from one Bombay blood group to another because of the presence of this third antibody, the anti-H antibody. So that's the Bombay blood group in short. But remember, it's the small h, recessive H gene responsible for this rare blood group. Landsteiner's law, second part, needs to be understood. The first part, the first sentence is, if a particular antigen is present on the red cell membrane of a person, then its corresponding antibody will be absent from the plasma of that particular individual. This is understandable, because if a particular antigen is present since birth, since the beginning in that particular individual, then it is considered as self protein or self antigen. and the body will not produce any antibodies against that particular antigen. So corresponding antibody will be absent from the plasma. This is understandable, easy to understand. If A antigen is present on the RBCs, then anti-A antibodies will not be present in the plasma of that individual. The second part needs to be understood. The second sentence of the Landstener's law is, if a particular antigen is not present on the RBCs in an individual, then the antibody corresponding to that antigen which is missing, the antibody corresponding to that would be present in the plasma of that individual. Now, why is that? From where this antibody has come? Let's understand this. A person's blood group is A, that means A antigen is present, so anti-A antibody will not be present in the plasma of that individual. Fair enough. But that means B antigen is absent on the RBCs of this individual. The corresponding antibody will be present in the plasma, means anti-B antibody will be present in the plasma of this individual. So A antigen and anti-B antibody in the plasma. The question is, from where this anti-B antibody came in the plasma of this individual? And the answer is, A and B antigens are present not only on the RBCs, I repeat, A and B antigens are present not only on the RBCs, but they are present elsewhere in the body and even outside the body in the nature. So, when a child is born, or due to the genotype, the child has got A antigen present on the RBCs. But immediately after the birth, the child will be exposed to both A and B antigens. As I mentioned, A and B antigens are also present in the nature, outside the body. So, not only on the RBC membranes, but elsewhere in the body and even outside the body, the A and B antigens are present. Now, this child with blood group A, with A antigen on the RBCs, will get exposed after birth through food, through bacteria, through milk. A and B, both antigens will enter the child's body. When A enters, it will be recognized as self and no antibodies will be produced against it because the child already has A antigen on the RBCs. But when B antigen enters from outside, the child's immune system will produce anti-B antibodies, and that is how anti-B antibodies are present in the A blood group individual. So remember, the reason is that A and B antigens are also present outside the body in nature, and therefore anti-A and anti-B antibodies are called as natural agglutinins, natural antibodies. And natural agglutinins are generally of IgM type of antibodies. Remember, the second part of the Landstener's law is not applicable to Rh blood group system. It is only applicable to the ABO blood group system. What about the Rh blood group? Anti-Rh antibody is not present normally or naturally in any individual. Rh positive individual or Rh negative individual, neither of them will have anti-Rh antibodies in the plasma. So, second part of the Landstener's law is not applicable to the Rh blood group system. It's only applicable to ABO. And why is that? That is because, now note this point, Rh antigen is present only and only on the RBC membrane, only and only on the red cell membrane. And therefore, imagine Rh positive individual. Right since birth, Rh antigen is present on the RBCs. So, this person will not produce anti-Rh antibodies in his own plasma. The immune apparatus will not form those antibodies. So, absent anti-Rh antibodies. What about Rh negative individual? Rh negative individual means Rh antigen is not present in this individual, and the person has never got exposed to Rh antigen. And therefore, this person also will not form anti-Rh antibodies in the plasma. So, anti-Rh antibodies are not natural agglutinins. They are called as induced agglutinins, and such agglutinins, such antibodies are of IgG type. So remember, the only way by which anti-Rh antibody can be found, can be generated or can be found in any individual is, note this point, when Rh negative individual is exposed to Rh positive blood. Because we mentioned this just now, and Rh antigen is present only in the blood, not anywhere else. So, the only possible exposure is this, that the Rh negative individual gets exposed to Rh positive blood. By mistake, blood transfusion has been given wrongly, or mother to fetus to mother. Such exposure happens, only then anti-Rh antibodies can be developed. Otherwise, they are not present, neither in Rh positive nor in Rh negative. So, Landstener's law, second part, not applicable here, that the Rh antigen absent, so anti-Rh antibody present in the plasma. No, that does not happen.